Welcome to the Sleep and Learn channel. We invite you to listen to interesting scientific facts that will help you fall asleep gently and, at the same time, learn something new. Relax, close your eyes, and let a calm voice guide you into the world of knowledge and sweet sleep. Understanding GMOs, definitions, origins, and foundations. Genetically modified organisms, commonly known as GMOs, are living beings whose genetic material has been altered using biotechnology. Unlike traditional breeding, which relies on selecting desirable traits over many generations, genetic modification allows scientists to introduce specific changes directly into an organism's DNA. These changes may involve enhancing existing genes, silencing others, or inserting genetic sequences from related, or sometimes unrelated species to achieve particular characteristics. The foundation of genetic modification lies in the structure of DNA, the molecular code that governs the development and function of all living organisms. DNA is composed of sequences of nucleotides arranged in genes, each carrying instructions for building proteins. Proteins, in turn, shape traits such as color, growth rate, resistance to disease, and tolerance to environmental conditions. By understanding and manipulating these sequences, scientists can influence how an organism develops and responds to its surroundings. Although the term GMO often evokes modern laboratory techniques, humans have been altering the genetics of crops and animals for thousands of years through selective breeding. Early farmers saved seeds from plants that exhibited favorable traits, gradually shaping species such as wheat, maize, and rice into forms very different from their wild ancestors. Genetic engineering represents a more precise continuation of this long history of agricultural innovation. Modern genetic engineering began in the 1970s with advances in recombinant DNA technology. Scientists discovered methods for cutting DNA at specific points and inserting new genetic material. This process often involves enzymes that function like molecular scissors and carriers known as vectors, such as plasmids, which deliver genetic material into cells. Once integrated, the modified DNA becomes part of the organism's genome and is passed on to future generations. One of the earliest widely adopted genetically modified crops was engineered to resist insect pests. By incorporating a gene from the soil bacterium Bacillus thuringiensis, certain crops gained the ability to produce a protein toxic to specific insects, but harmless to humans and most other organisms. This innovation reduced the need for some chemical insecticides, illustrating how targeted genetic changes can influence agricultural practices. Another common modification involves herbicide tolerance. Crops engineered with this trait can survive the application of specific herbicides, allowing farmers to manage weeds more efficiently. Such traits are designed to improve crop yield stability and reduce competition for nutrients and sunlight. It is important to distinguish between different categories of genetic modification. Some changes introduce traits already present within the same species, while others involve transferring genes between different organisms. Recent developments, including gene editing tools such as CRISPR, enable even more precise alterations without necessarily introducing foreign DNA. These techniques blur traditional definitions and expand the possibilities of biotechnology. The development and approval of genetically modified organisms typically involve extensive testing and regulatory oversight. Safety assessments examine potential impacts on human health, environmental interactions, and biodiversity. Regulatory frameworks vary by country, reflecting differences in scientific evaluation, policy approaches, and public perception. From a global perspective, genetically modified crops are cultivated in many regions, particularly for commodities such as soybeans, maize, cotton, and canola. Their adoption often correlates with agricultural scale and economic considerations. At the same time, debates surrounding GMOs continue in scientific, ethical, and social contexts, emphasizing the importance of transparency and informed decision-making. When viewed through an encyclopedic lens, GMOs represent a convergence of molecular biology, agriculture, economics, and public policy. They illustrate humanity's increasing capacity to understand and influence the genetic foundations of life. This capacity, while powerful, operates within complex ecological and societal systems that require careful study and responsible stewardship. The science behind genetic modification and how it works. Genetic modification is rooted in molecular biology, the study of how genes function at the level of DNA, RNA, and proteins. To understand how genetic modification works, one must first consider how cells interpret genetic information. Within each cell, DNA serves as a stable archive of instructions. When a particular gene is needed, it is transcribed into messenger RNA, mRNA, which carries the code to cellular structures called ribosomes. There, proteins are assembled according to the genetic blueprint. By altering DNA sequences, scientists influence which proteins are produced and how they function. The process of genetic modification typically begins with identifying a gene associated with a desired trait. This trait may involve resistance to pests, tolerance to environmental stress, enhanced nutritional content, or improved storage life. Once identified, the gene is isolated using enzymes that can cut DNA at precise locations. These enzymes, often called restriction enzymes, recognize specific nucleotide sequences and act as molecular tools for editing genetic material. After isolation, the selected gene must be inserted into the target organism. In plants, one common method involves the bacterium Agrobacterium tumefaciens. This naturally occurring soil bacterium has the ability to transfer part of its DNA into plant cells. Scientists harness this capability by replacing the bacterium's native genetic material with the gene of interest. When the bacterium interacts with plant cells, it delivers the engineered DNA into the plant genome, where it becomes integrated and stable. Another method, sometimes called the gene gun technique, physically introduces microscopic particles coated with DNA into plant cells. These particles penetrate the cell wall and membrane, allowing the new genetic material to enter the nucleus. Though more mechanical than biological methods, this approach can be effective for species less responsive to bacterial transfer. Once the gene is incorporated, cells are cultivated in controlled laboratory conditions to regenerate into full plants. Researchers then test these plants to confirm that the inserted gene functions as intended. This involves evaluating protein expression, stability across generations, and absence of unintended effects. Only after extensive laboratory and field trials can a genetically modified organism proceed toward regulatory review. In animals and microorganisms, similar principles apply, though techniques may vary. Microbial genetic engineering is widely used in medicine. For example, certain bacteria are engineered to produce human insulin, replacing earlier extraction methods that relied on animal sources. This application demonstrates how genetic modification extends beyond agriculture into pharmaceutical production and industrial biotechnology. Recent advancements in gene editing have refined the precision of modification. Technologies such as CRISPR-Cas systems enable scientists to make targeted changes directly within an organism's genome without necessarily introducing foreign DNA. CRISPR functions like a guided molecular scalpel directed by a short RNA sequence that matches a specific DNA region. The Cas enzyme cuts at the designated site, allowing genes to be removed, altered, or replaced with exceptional accuracy. The distinction between traditional transgenic modification and gene editing
has become increasingly relevant in scientific and regulatory discussions. While transgenic methods may introduce genes from unrelated species, gene editing can adjust an organism's existing genes in ways that mimic natural mutations, though achieved with greater speed and predictability. Despite its technical complexity, the fundamental goal of genetic modification is relatively straightforward: to modify biological instructions in order to achieve specific, beneficial traits. Each step, from gene discovery to organism development, involves layers of testing, verification, and monitoring. The scientific rigor underlying these processes reflects the importance of ensuring safety and reliability. Viewed calmly and systematically, genetic modification is not a sudden departure from biological principles, but an extension of humanity's expanding knowledge of genetics. It operates at the microscopic scale within the universal language of DNA, where life's diversity originates and evolves.
Applications of GMOs in agriculture and medicine. The practical applications of genetically modified organisms extend across agriculture, medicine, and industrial biotechnology. While the scientific mechanisms behind genetic modification operate at the microscopic level, their effects are often visible in fields, hospitals, and laboratories around the world. These applications illustrate how targeted genetic changes can influence food production, disease treatment, and manufacturing processes. In agriculture, one of the primary goals of genetic modification is crop protection. Plants engineered to resist insect pests reduce crop losses and may decrease reliance on certain chemical insecticides. By incorporating genes that produce specific protective proteins, these crops can defend themselves against targeted insects without affecting most beneficial organisms. This precision aims to improve efficiency while maintaining ecological balance within agricultural systems. Herbicide-tolerant crops represent another widely adopted application. By enabling crops to survive specific weed control treatments, farmers can manage competing plants more effectively. This approach simplifies field management and can support conservation tillage practices, which reduce soil erosion and preserve moisture. In some regions, such practices contribute to improved soil structure and lower fuel consumption in farming operations. Beyond pest and weed management, genetic modification also addresses nutritional enhancement. Certain crops have been engineered to contain increased levels of essential vitamins or minerals. A well-known example is vitamin A-enriched rice, developed to help address nutrient deficiencies in areas where rice is a dietary staple. Such innovations aim to complement broader public health strategies by integrating nutrition directly into staple foods. Genetically modified crops can also improve tolerance to environmental stress. Traits related to drought resistance, salinity tolerance, or temperature adaptability are under continuous development. As climate variability increases, these characteristics may help stabilize yields in regions vulnerable to extreme weather conditions. In medicine, the impact of genetic engineering is both long-standing and profound. One of the earliest and most transformative achievements was the production of human insulin using genetically engineered bacteria. Prior to this innovation, insulin was extracted from animal pancreases. The recombinant approach allowed for large-scale, consistent production of insulin identical to the human form, significantly improving treatment for people with diabetes. Beyond insulin, genetic engineering supports the production of vaccines, clotting factors, growth hormones, and monoclonal antibodies. Many modern vaccines rely on recombinant DNA techniques to produce specific viral proteins that stimulate immune responses without causing disease. These technologies enable more targeted and efficient pharmaceutical development. Gene therapy represents another frontier in medical biotechnology. Rather than modifying crops or microorganisms, gene therapy seeks to correct or compensate for faulty genes within human cells. By delivering functional genetic material to specific tissues, researchers aim to address inherited disorders at their molecular source. While still advancing, this field reflects the expanding possibilities of genetic science. Microorganisms engineered for biotechnology also contribute to industrial processes. Genetically modified yeast and bacteria are used to produce enzymes for food processing, biofuels, and biodegradable materials. These applications often operate behind the scenes, yet they influence daily life through products ranging from cheese to renewable energy sources. The scope of GMO applications demonstrates the versatility of genetic tools. From enhancing crop resilience to manufacturing life-saving medicines, genetic modification bridges agriculture and healthcare. Each application undergoes regulatory review and scientific evaluation, reflecting the responsibility inherent in altering living systems. Viewed in a measured and analytical way, GMOs represent not a single technology, but a spectrum of applications rooted in molecular precision. Their influence extends quietly, yet significantly, through global food systems and medical practice. Environmental considerations and ecological interactions. The environmental dimension of genetically modified organisms extends beyond laboratories and crop fields into broader ecological systems. Because agriculture interacts directly with soil, water, insects, and surrounding plant life, the introduction of genetically modified crops requires careful evaluation of ecological relationships. These considerations form a central part of scientific and regulatory assessment. One important topic involves gene flow, the movement of genetic material from cultivated crops to related wild species. Pollen can travel through wind or insect activity, potentially transferring traits such as herbicide tolerance to compatible plants. In regions where wild relatives are present, monitoring and management strategies help reduce unintended cross-pollination. Buffer zones, crop rotation, and controlled planting schedules are among the approaches used to minimize ecological overlap. The impact of genetically modified crops on non-target organisms is another area of study. Crops engineered for insect resistance are designed to affect specific pest species. Scientists evaluate whether beneficial insects, such as pollinators or natural predators, experience unintended effects. Long-term ecological studies assess biodiversity within and around GMO cultivation areas, comparing them to conventional agricultural systems. Soil ecosystems also play a role in environmental analysis. Beneath the surface, communities of bacteria, fungi, and invertebrates contribute to nutrient cycling and plant health. Research examines whether genetically modified crops influence these microbial networks through root exudates or crop residue. Current findings generally indicate that soil dynamics are influenced more strongly by farming practices than by genetic modification itself, though ongoing study remains essential. Herbicide-tolerant crops introduce a distinct ecological dynamic. By simplifying weed management, they can support conservation tillage, which reduces soil erosion and carbon loss. However, repeated reliance on a single herbicide may contribute to the evolution of herbicide-resistant weeds. This phenomenon reflects a broader biological principle. Populations adapt to selective pressures over time. Integrated weed management strategies, combining multiple approaches, are therefore recommended to maintain long-term sustainability. Biodiversity within agricultural landscapes depends not only on crop genetics, but also on land use patterns. Large-scale monocultures, whether genetically modified or conventional, can reduce habitat diversity. Conversely, diversified farming systems and ecological corridors can support wildlife alongside cultivated crops. In this context, environmental impact arises from management choices as much as from genetic traits. Water systems are another consideration. Agricultural runoff can carry nutrients or residues into nearby rivers and lakes. Monitoring programs evaluate how different crop systems influence water quality. Sustainable irrigation practices and careful input management contribute to reducing ecological pressure on aquatic ecosystems. From a broader ecological perspective, genetic modification may also offer environmental benefits. Crops requiring fewer chemical applications can reduce certain types of agricultural emissions. Drought-tolerant varieties may help conserve water resources in arid regions. Such potential advantages must be weighed alongside possible risks, emphasizing balanced evaluation rather than singular conclusions. Ecosystems are complex networks of interactions shaped by climate, species diversity, and human activity. The introduction of genetically modified organisms becomes one factor within this larger system. Scientific assessments aim to understand how that factor interacts with existing ecological processes. Ultimately, environmental considerations surrounding GMOs illustrate the interconnectedness of biology and landscape management. The ecological impact of genetic modification depends not solely on the presence of a modified gene, but on how agricultural systems are designed, monitored, and adapted over time. The future of biotechnology and emerging innovations. Biotechnology continues to evolve at a pace shaped by advances in genetics, computational biology, and engineering. While early genetic modification focused primarily on introducing specific traits into crops or microorganisms, emerging innovations expand the scope of what is possible. The future of biotechnology lies not only in altering genes, but in understanding complex biological systems as interconnected networks. One of the most transformative developments is precision gene editing. Tools such as CRISPR-based systems allow scientists to modify DNA with extraordinary accuracy. Rather than inserting entire genes from external sources, researchers can adjust existing genetic sequences, deactivate problematic genes, or enhance natural traits. This level of precision reduces unintended changes and accelerates research timelines. Synthetic biology represents another frontier. Unlike traditional genetic engineering, which modifies existing organisms, synthetic biology designs and constructs new biological components or pathways. Scientists can assemble genetic circuits that instruct cells to perform novel functions, such as producing biodegradable materials or capturing carbon dioxide. In this field, biology and engineering increasingly converge. Agricultural innovation is also expanding beyond single gene traits. Researchers are exploring polygenic traits, characteristics controlled by multiple genes, to improve yield stability, nutrient efficiency, and climate resilience. Advanced genomic analysis enables the identification of complex trait patterns, supporting more sophisticated breeding strategies that integrate biotechnology with conventional methods. In medicine, personalized therapies are gaining prominence. Advances in genomic sequencing allow treatments to be tailored to an individual's genetic profile. Gene editing techniques are being investigated for correcting inherited disorders at their source, while engineered immune cells are designed to recognize and combat specific diseases. These applications reflect the growing integration of biotechnology and precision medicine. Another emerging area involves microbiome research. The communities of microorganisms inhabiting soil, plants, animals, and humans play critical roles in health and ecosystem function. Biotechnology may enable targeted adjustments to microbial populations, improving crop productivity or supporting human health through carefully designed probiotic interventions. Environmental biotechnology also holds potential for sustainability. Engineered microorganisms are being developed to break down pollutants, convert waste into bioenergy, or assist in soil restoration. Such approaches aim to address environmental challenges through biological processes rather than purely chemical or mechanical solutions. Digital technologies increasingly support biotechnology research. Artificial intelligence and machine learning assist in analyzing vast genomic datasets, predicting protein structures, and modeling complex biological interactions. Computational tools accelerate discovery by identifying promising genetic targets before laboratory experimentation begins. Despite these innovations, careful oversight remains essential. As technologies become more powerful, ethical frameworks and regulatory systems must evolve accordingly. International collaboration and transparent communication will continue to guide responsible implementation. The trajectory of biotechnology suggests a shift from isolated modifications toward systems-level understanding. Rather than altering a single trait in isolation, future research may consider entire networks of genes, environmental interactions, and ecological outcomes. This holistic perspective reflects a maturing field that recognizes the complexity of living systems. In a calm and measured view, the future of biotechnology is neither utopian nor cautionary by default. It is shaped by scientific progress, policy decisions, and societal values. As knowledge deepens, the tools for influencing biological systems become more refined, opening new possibilities for agriculture, medicine, and environmental stewardship. The story of genetic modification thus becomes part of a broader narrative, one in which humanity continues to explore the language of DNA with increasing precision. Whether applied to food systems, healthcare, or sustainability, biotechnology remains a dynamic field guided by discovery, responsibility, and thoughtful innovation.